Speak Student!
Molecular genetics. Cloning. A la Shmoop.
Okay, we're on next lesson, DNA technology with Dr. Ruth Tenen. So Ruth, tell us, what does cloning mean when we're talking about genes? Like, we hear about cloning all the time in the press, and there's like frogs and sheep and congressmen who've all been cloned. How does cloning work? Like, run us through like what that means.
Yeah, so cloning in the context of DNA basically means isolating a piece of DNA or a gene from an organism and then putting it into another context, like into another piece of DNA or into another organism in many copies.
But so talk to us, how do we isolate a gene? Like what is, that sounds very technical.
Yeah, so the basics of doing that is, like, let's say you've got your chromosome and there's a gene that's somewhere on there. There are special enzymes called restriction enzymes that can chop out the gene of interest.
And we can manipulate the restriction enzyme. I mean, we can actually inject that into a gene and ourselves like pull out that code?
Yeah, it's basically you isolate the DNA from whatever source you want, and then you just add the enzyme in a tube and it does its thing. Enzymes are derived from bacteria, so we didn't actually make them in most cases, but we figured out what they do.
Wow, cool. Okay, so we've pulled out that isolated gene, and then what do we do with it? Like what happens from it?
What are plasmids?
Yeah, so usually before you can do anything, you have to put it into, so a gene by itself can't do much. You usually have to put it into some sort of carrier or vector. And so in most cases, this is called a plasmid, which is basically a circular piece of DNA that you can put other DNA into. So you can stick your gene of interest into this plasmid, and then you can do cool stuff with your plasmid, like put it into bacteria or put it into other cells and kind of study the function of your gene.
How does cloning work? How do we isolate a gene? What are plasmids?